Let's move on to another category of antibacterial agents, the bacterial protein synthesis inhibitors. So the bacterial protein synthesis inhibitors are narrow-spectrum agents. They act either on the 50S subunit, such as linezolid or streptogramins or glycosamides, or they're broad-spectrum agents that are like the macrolides and chloramphenicol. Other broad-spectrum agents that act on the 30S subunit include the tetracyclines, the aminoglycosides. Now let's just quickly do some definitions because we need to do this before we go on. There's something called a post-antibiotic effect. So this is an anti-infective effect that lasts after the elimination of the antibiotic from the body. That's often because we have some kind of I wouldn't say permanent, but more long-lasting effect, in this case on the 50 or 30S subunits. Bactericidal means that it kills bacteria. Bacteriostatic means that it just stops the bacteria from replicating but doesn't kill them. So the bacteria are still alive, but the numbers aren't increasing, so the other immune-mediated killing of the bacteria can occur unimpeded. The 70S ribosomal mRNA subunit is composed of a 50S and a 30S. Okay, I know. 50 plus 30 is 80, and how can 50 plus 30 equal 70? Don't worry about it. The point is that the 50S and the 30S make up a 70S unit. Time-dependent agents. So these are drugs that have increased killing activity with time. This is different from concentration-dependent killing, where the drugs have increased killing activity with concentration. So there you have some basic definitions, and we're going to be using them as we go forward in this lecture. Let's go on to the 70S ribosomal unit. The 70S ribosomal unit is made up of the 50S and the 30S units. Now, this is a hamburger. Think of it as a hamburger bun. So the 50S is the top of the bun and the 30S is the bottom of the bun. So the 30S is smaller and flatter, the 50S is bigger and puffier. Now, how this works is that you have this charged transfer tRNA on the end there, bringing in the 7th amino acid. You can see the little 7 there. That's an amino acid. So the charged transfer RNA comes in and sits on top of the mRNA or messenger RNA. And the messenger RNA is the base that determines what the coding is going to be for the protein. Let's pretend for a moment that this particular protein is, a protein involved in metabolism. There's a specific code that's coded by mRNA, and it tells the transfer RNA which amino acids to bring in. As it goes through the hamburger, it's as if you had a slice of cheese being pulled through the hamburger buns. You get different coding, and you have a very specific sequence that makes up the protein. So the protein is the sort of the chain of blue circles there, kind of like a stringy onion coming out of the hamburger. So that protein is very specifically configured based on the characteristics of the messenger RNA. Now, the uncharged transfer RNA is then discarded and eventually becomes charged, and then it grabs another amino acid. Now, the drugs that we're going to talk about act at different points in this 70S ribosomal unit. So, for example, chloramphenicol blocks the transpeptidation, which is the joining of two amino acids. The macrolides also block transpeptidation, but in a slightly different area. The tetracyclines bind to the 30S subunit and prevent binding of incoming transfer RNA. So they actually stop, transfer RNA number 7 from joining. Linezolid has a unique site that inhibits initiation complex formation. So initiation complex is the very first reaction that brings the 30S and the 50S together and allows everything to start working. On the other end of the production line there, the streptogramins block exit ports for polypeptides, so new ones can't come in and overall transcription is inhibited.